Hi, I'm Dr. David Dobson. Welcome to Conversations. Today, my guest is Folefac Charlemagne Asonganyi. Charlemagne is a PhD researcher at the Department of Oncology at Aarhus University in Denmark. Today, we will discuss his research on experimental clinical oncology. It's a great pleasure to have you with me today, Charlemagne.
Thank you, Dr. Dobson. I hope we are going to have a good conversation and your audience will be happy to get what we are going to discuss.
Thank you. What is experimental oncology?
I think for me to really give you a true definition of experimental oncology, we have to first of all understand what oncology is all about. So oncology is a branch in medicine that has to do with how we can diagnose, how we can treat, or how we can prevent cancer. And I would say that it's a collective area of study and practice with a one goal to address issues relating to cancer. I use the word collective because we have different professionals working within the field. For example, we have medical doctors who have studied and then specialized on how to treat cancer. We call them oncologists. Okay. We have surgeons, those who are specialized in operating and taking out the cancer from patients. We have radiologists, those who are specialized in using radiation to treat cancer. Yeah. We have pathologists, those who are specialized in analyzing samples to tell if it has cancer or not. Mm-hmm. We have biologists like myself, those who are understanding studying and understanding cancer, and then maybe trying to come out with research on how to develop new drugs or new treatment strategy. Okay. And within oncology, we have various departments, and one of the departments which I would like to talk about now is the experimental oncology, which has to do with designing experiments, which could be in vivo experiments involving doing the experiments on experimental animals, or in vitro experiments where we are doing the experiments out of the body of animals. And the ultimate goal is to understand cancer biology, to see how we can come out with new drugs which we can treat the cancer, or we can use this drug to prevent, and also to see how we can improve on already existing cancer treatments. And so the importance of oncology cannot be overemphasized, but there are so many importance of experimental oncology experiment. But I'm going to mention just a few to you. For example, from the result that we get from conducting experiments, we can pave way for new treatments because we know what is happening. Also, it opens rooms for personalized treatment. For example, cancer is a genetic disease. So in doing experiment, we know how the genes are formed. And so we can come out with a drug such that we can give a particular drug to specific patients. It also opens route for us to be able to compare between existing drugs to know which drug is working very much and also which ones are not more working. Then I would say that it opens doors for clinical trials. Because when we come out with new drugs, for us to take it to the patients, we need to first of all test it on animals. And so it all starts on experimental oncology. And so the general goal of experimental oncology is to see how we can advance in cell biology, in cancer biology.
So it's very fascinating and look like a very collaborative approach with a lot of partners are involved in. So it's not just one area, but several departments, several expertise, and several people involved in this interesting research. And you're coming from a biology perspective. So what are you studying for your PhD?
I think I'll be a little bit elaborate. So in my PhD, I'm using animal and tumor models to see how we can improve on the treatment of cancer. To see how we can improve on radiation treatment of cancer. So, when cancer develops, it grows, and then there comes a time when the cancer will develop their own blood vessels. And then when they develop their own blood vessels, the blood vessels are different from the normal blood vessels in other healthy cells within the body. And so that creates a problem because they cannot provide the cancer with enough oxygen, enough nutrient, and so it creates a very difficult environment.
Okay.
But over the years, we have different approaches, different treatment options, which they use to treat cancer. But one of those options, which I would say is like the second best option, is radiation.
Okay.
Because you have more than close to 50% of cancer patients receiving radiation as a treatment at each point.
Yes.
But the use of radiation is limited because for radiation to work so well, we need oxygen.
Yeah. Yeah.
But because we have that cancer cell population which lack oxygen, it makes it that if we want to kill more cancer cells, then we need to use higher radiation dose. But if we use higher radiation dose, we end up killing healthy tissues. So the goal is how can we improve on radiation such that we will not administer higher radiation. And one strategy is to use hyperthermia, which is just is to combine radiation with hyperthermia. And in oncology, when we talk about hyperthermia, we mean heating specific tissues to about temperature within the range of 40 to 45 degrees. So temperature within that range.
Okay.
And so when we use a temperature within that range, it has a lot of advantages that we can use to combine with radiation. For example, if you administer the temperature before bringing the radiation, it opens the blood vessels. It brings more oxygen. And so if you bring radiation, it means you can make more oxygen available at the time of treating the cancer.
Okay.
So it means at the end of the day, we will reduce the radiation dose that we need. And then secondly, when you treat cancer, we try to destroy the DNA of cancer cells.
Oh, okay.
And a cell will die only when you have destroyed the DNA completely. And the cells have a strategy where when the DNA is destroyed, they try to repair it.
Okay.
So it means that if we use radiation to hit and destroy the DNA of cancer cells, they also try to repair. And one way for us to stop the cells from repairing their DNA is to bring temperature within the range of 40 to 45, because it's going to stop the cancer cells from repairing their DNA.
Yes. Yeah.
Then another advantage of using hyperthermia is that hyperthermia temperature can specifically kill cancer cells which lack oxygen. But for us to know how to combine hyperthermia and radiation, we need to optimize treatment conditions. We need to know the time to bring in the temperature.
Yeah.
This is not something new. It's something which they have done it before. Some people have done it.
Yeah.
But from previous studies, we didn't have a specific study where we have tested this different temperature, different time interval between applying radiation and heat. So as far as my project is concerned, I'm using this animal and tumor models to see how I can define a time interval between applying radiation and heat.
Yeah.
To look at the best time interval to know the minimum radiation dose that we need. That is going to give us the optimum result. I'm also looking at how we can try to assess toxicity, because we cannot translate the result to the clinic if it is not safe for our patients. So I'm also using this condition to see if they are toxic or not, or to see if they are safe for us to translate it to the clinic. So that is just an overview of what I'm doing as far as my PhD is concerned.
You're doing a great work. Sounds so important and so exciting. So what have you learned so far from your research?
I think I've learned a lot. And I can just give you a few of those ones. I have learned that hyperthermia indeed is a good sensitizer or enhancer of radiation. So that is one thing which is certain. It's not new knowledge, but it's already, I'm just adding to already what is known. Then I've learned that a single hyperthermia temperature is enough if we apply it correctly to enhance radiation. Because what I've learned with my story that when I give 3 fractions of radiation, then at the end of the day, I give 1 heat treatment. So what I've learned is that a single heat treatment, which is given effectively, can help improve on radiation treatment.
Right. So for your experiment and all this trial, are you using actual subjects for your experiment, or how are you conducting these experiments? On whom are
So
you conducting experiments?
I'm conducting the experiment on animals. So what I do is I'm using some animal, they call them CDF mice, which are experimental animals.
Okay.
So what I do is that I induce cancer on them. So the cancer model that I'm using is a breast cancer.
All right. Okay.
So I induce it on them. Then I use radiation to treat them. I use now different radiation doses. The doses that I'm currently using are 5 gray, 10 gray, and 15 gray. But I'm giving 3 fractions over 1 week.
Okay.
Then at the last day, after I have given the last fraction, then I test the different time interval. I have groups that I test after 30 minutes. I test after 90 minutes or after 180 minutes.
Okay.
Then I use what we call the tumor delay growth assay. So and then I monitor the animals on daily basis to see if the tumors are growing or not.
Okay. Nice.
Then I monitor the animals for 90 days. Then I draw conclusion. Maybe I have tumor control, or I have tumor delay growth, or it did not work at all.
Okay. So you're probably spending a lot of time in the lab, I guess, for these experiments.
I mean, when you are with animals, you have to, because the animals have to be in good conditions. You have to make sure they are under experimental condition. They are feeling very well. They are not sick.
Yes. So how far are you trying your experiment on the human trials?
In human trials, for us to take it to the human trial, we have to first of all, make sure that all these conditions are okay. So the first thing is for me to make sure that I can treat tumor with this condition. Then I test them now I test for toxicity on animals.
Okay.
And until this condition, they are working on animals and they are safe on animals, then can we translate it to the clinic? And like I mentioned, the use of hyperthermia and radiation is not something new. What I'm doing is not new. But we have very few centers which are using it.
Okay.
But I'm just trying to see how we can optimize the treatment conditions.
Okay.
Yes.
Okay. So how would your research possibly benefit cancer patients?
There are a lot of ways in which the cancer patients will benefit from this research. And just like I already mentioned, if we successfully apply hyperthermia, we reduce radiation doses that we are applying. If we are reducing radiation doses, it means we are improving the quality of life of cancer patients, because it's not just to treat the patient and cure the cancer, but to make sure that at the end of the day, the quality of the life of cancer patients is maintained or is improved. So that is one way in which the result will benefit the cancer patient. Then in my study, I'm using different sources of radiations. I'm using both photon radiation and proton radiation. And I'm applying them stereotactically. So I'm applying them with higher dose per fraction. And with the higher dose per fraction, we can kill more cancer cells. So it means that the number of days that the patient will have to go to the hospital, if they were receiving conventional treatment where they are giving smaller doses, maybe they have to go to the hospital like 30 days for them to have a complete dose. But with the approach that we are using, we see that if it works, then at the end of the day, the patient will not have to go to the hospital as many times. So we are not just reducing the workload to the doctor. We are also making it comfortable for the patient to receive the treatment.
So what is your advice to future researchers who wish to continue your research topic?
My advice will be in 2 phases. If you want to continue directly with the project that I am working on, because at the moment I told you I'm working on breast cancer model. Then I will advise others to try other models, because you have different cancer models to try on other models. But I do believe that if my treatment work on solid cancer, then it can cure any other cancer. But before drawing that conclusion, it would be nice that they test it on other models. Also, one challenge that my project faces is that to deliver the heat on the tumor is very challenging. So that would be an area where I would advise somebody to see how we can explore, to see how we can apply the heat successfully. So that is directly linked to my project. But of course, if you really want to come into my project or to work on cancer, It's a very tedious area. You should be ready to collaborate with other researchers who are in the field. You should be ready to maybe go to scientific meetings where you will meet people, do presentations.
Yeah.
You should be able to bring your results to the public, like we are discussing, so that people know what you're doing. You can have one or two advice.
Yes.
You should be open to different changes, because the treatment that we are, the drug that we are using today to treat cancer might change in the future, because it's a disease of the genes. We keep having mutation, so the drugs may change.
Yeah.
So you should be ready to adapt for changes.
Yeah. Yeah. So, Charlemagne, what's next for you? You almost finishing your PhD.
Yes, I'm almost finishing my PhD, and my academic journey is not like I need to move from the PhD to postdoc, or from postdoc to a professor.
Yeah.
Yes, I always try to take it one step at a time. And at the moment, my main focus is to make sure I finish it effectively.
Yes.
But of course, the future is open. There are a lot of areas where I can go into.
Yeah.
But it doesn't matter. I know I will remain a researcher. Once a researcher, you remain a researcher. It doesn't matter if I decide to go to the industry to work.
Yeah.
But I know that in one way or the other, I'll be contributing to this field of research.
Nice. Thank you for your time, Charlemagne, and all the best in completing your PhD.
Thank you so much for inviting me on your platform. And also, I would like to use this opportunity to thank the sponsors of my PhD, because this PhD is one of European project known as the HyperBoost project, which brought together a group of scientists so that we can see how we can improve on the use of hyperthermia in combination with radiation to treat cancer. So I would like to express my appreciation to them. I'm going to give you their website so that you can put it on your page, so that people can have a look at what is going on there. Also, I would like to acknowledge my supervisor, Professor Mike Hosman, who is providing me with all the guidance, with what I need. And also this project, I'm collaborating with a colleague who is Priyanshu. We are working on the same project. So, and our technician and any other person who have assisted me, whom the name is not mentioned here. And most importantly, I want to thank you for inviting me here. And I hope that this is not the last time we'll be having this conversation.
Of course. Love to have you back again, Charlemagne. Thanks again.